Immunology Insights, Basics and Beyond, Chapter 60, Adoptive Cell Transfer Therapies Beyond CAR T Cells, TCR, TIL and NK cell approaches. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored next-generation checkpoint therapies and how combination immunotherapy tries to outsmart tumors by targeting several inhibitory pathways at once. Today, we stay in the world of cancer immunotherapy, but shift from drugs that modulate the immune responses to therapies that physically add immune cells back into the patient. We have already discussed CAR T cells as one of the great success stories in hematologic malignancies. But CAR T cells are only one branch of a broader family called adoptive cell transfer, or ACT. In this chapter, we will look beyond CAR T cells and explore three other important ACT strategies: TCR-engineered T cells, tumor-infiltrating lymphocyte or TIL therapy, and natural killer or NK cell therapies. Each of these approaches comes with its own strengths, limitations and clinical niche. So let's talk science. So what is adoptive cell transfer? Adoptive cell transfer means taking immune cells out of a patient or a donor, manipulating or expanding them in the lab, and then reinfusing them to provide a boosted tumor-specific immune response. Two key dimensions run through all ACT approaches. First, autologous versus allogeneic. Autologous means that the cells come from the patients themselves. They are naturally compatible, but often exhausted or low in numbers, and production takes weeks. This makes them immunologically safe but slow and sometimes less potent, especially in heavily pre-treated patients. And then we have allogeneic, where cells come from a healthy donor. They can be banked off the shelf and given quickly, but they must be engineered to avoid rejection or graft versus host disease, or GVHD. The trade-off is speed and scalability versus a higher need for genetic engineering and immune control. As second dimension, we have ex vivo versus in vivo engineering. Ex vivo means that cells are collected from patients or donors, modified in the lab, and then reinfused, which is the current standard for CAR T, TCR-T, TILs, and many NK products. This allows precise quality control and characterization, but it is complex, expensive, and logistically demanding. And then we have the in vivo approach, which is in development. Vectors or nanoparticles deliver genetic instructions directly into the immune cells inside the patient's body, turning them into therapeutic cells without ever taking them out. In principle, this could be faster and cheaper, but it is harder to control which cells are modified and for how long, which raises safety and targeting challenges. Another central concept is the type of tumor antigens the cells recognized. We have tumor-associated antigens or TAAs. They are proteins overexpressed on tumors, but also present at lower levels on some normal tissue. For example, HER2 or WT1. They are easier to find and validate, but carry a risk of on-target off-tumor toxicity when normal cells are hit. We have specific tumor antigens or neoantigens, which are truly new peptides, often derived from somatic mutations expressed only by cancer cells. These are ideal targets because they minimize collateral damage, but identifying and validating them is technically demanding and often patient-specific. We have viral antigens. In virus-driven cancers like HPV, EBV, or HBV, viral proteins are excellent non-self targets. They offer clear discrimination between infected tumor cells and healthy tissues, but are limited to cancers with a viral etiology. Finally, we need to recall also MHC restriction. Conventional T cells recognize short peptides presented by MHC, or HLA molecules, on cells. A TCR recognizes a specific peptide-MHC combination, as we already learned in an earlier chapter. This makes TCR-based therapies exquisitely specific, but also means they only work in patients with the right HLA type and the right antigen, and tumors can escape by downregulating MHC. By contrast, NK cells sense stress and missing self, so the loss of MHC1, and are not MHC restricted, giving them a complementary role and make them ideal candidates for tumors that downregulate MHC. Chimeric antigen receptor or CAR T cells also bypass MHC by binding surface antigens directly with their CAR, which usually has an engineered antibody component, which broadens eligibility but focuses targeting on extracellular proteins only. Solid tumors pose an extra challenge. Poor penetration, heterogeneous antigens, hostile microenvironments, whereas blood cancers tend to be more accessible and antigen uniform. New ACT strategies are being developed for both, but they behave quite differently in these two worlds. Well, now let's have a look at the three major new approaches. First, TCR-engineered T cells, reprogramming recognition from the inside. CAR T cells recognize intact antigens on the cell surface via an antibody-derived receptor. TCR-engineered T cells, or simply TCR T cells, in contrast, go one step deeper. They recognize peptides that were generated from proteins inside the cells and then presented on MHC or HLA molecules on the cell surface. This allows them to target intracellular proteins, including many neoantigens and cancer testis antigens, such as MAGE-A4 or NY-ESO-1, that never appear as intact surface molecules, only as peptide MHC complexes. In this way, TCR T cells can, unlike CAR T cells, access a much broader universe of intracellular tumor antigens, an attractive feature especially for solid tumors, such as synovial sarcoma or certain melanomas, where suitable surface targets are scarce. So, just to recap, what is a TCR and why replace it? The T cell receptor or TCR is essentially the eyes of a T cell. Each T cell naturally expresses its own TCR, tuned to recognize one very specific peptide MHC combination. In TCR-T therapy, we take T cells from a patient and add or replace with a new TCR that has higher affinity and well-defined specificity for a tumor antigen presented by a particular HLA type. For example, HLA-A*0201, presenting a peptide from NY-ESO-1. This makes the engineered T cells extremely specific for tumor cells that express both the antigen and the matching HLA molecule. But where do these therapeutic TCRs then come from? Well, sometimes from rare patients or donors who naturally mount strong T cell responses against a given tumor or viral antigen. For example, spontaneous T cell responses to NY-ESO-1 in certain cancers. Sometimes from immunized donors or humanized mice engineered to express human HLA molecules, which are then vaccinated with the target antigen. The TCR genes are cloned, sometimes affinity optimized in vitro, and then introduced into patient T cells using viral vectors or other gene transfer methods. Once infused back into the patient, TCR T cells patrol the body, scan peptide MHC complexes on cells, and selectively kill those that display their cognate tumor peptide. They use the same effector machinery as natural cytotoxic T cells, perforin and granzymes to induce apoptosis, and cytokines such as interferon gamma and TNF-alpha to shape the local immune response and recruit additional immune cells. This makes them conceptually similar to physiological anti-tumor T cells, but with a much sharper lens, so to say, and a higher precursor frequency. A major milestone was reached in 2024 when the FDA approved afami-tresgene autoleucel, or Tecelra, a TCR therapy targeting the cancer testis antigen MAGE-A4 for metastatic synovial sarcoma, marking the first approved TCR product in oncology and a proof of concept that this strategy can work in solid tumors. However, there are important limitations. TCR T cells are both HLA restricted and antigen specific. Only patients with the right HLA type and sufficient expression of the target antigen are eligible. In addition, because TCRs recognize short peptides, there is a theoretical risk of off-target recognition of similar peptides on healthy tissues, which can lead to serious toxicity. Compared to CAR T cells, TCR Ts gain access to intracellular antigens and may be better suited for certain solid tumors, but they pay for this with HLA restriction, more complex patient selection, and a narrower therapeutic window that demands very careful target validation. Second, we have tumor-infiltrating lymphocytes or TIL therapy, amplifying what's already working. Tumor-infiltrating lymphocyte or TIL therapy builds on a simple observation. If lymphocytes have already entered a tumor, they have found it. There are just so few and too exhausted to actually clear it. TILs are immune cells that reside inside the tumor tissue. In practice, the therapeutically relevant cells are mainly T lymphocyte, so CD8 positive cytotoxic and CD4 positive helper T cells, alongside some additional lymphocytes like B cells, NK cells, and NKT cells. They have already seen tumor antigens in vivo, including many patient-specific neoantigens, and are therefore highly specific and valuable. However, chronic overstimulation, checkpoint pathways, and an immunosuppressive microenvironment progressively blunt their function. TILs are often partially exhausted, expressing high levels of inhibitory receptors like PD-1, LAG-3, and TIM-3, and are suppressed by local factors like TGF-beta, IL-10, regulatory T cells, and myeloid-derived suppressor cells. Also, these connections were discussed in earlier chapters of this podcast. Well, this illustrates that the immune system tries to attack the tumor, but is held back and eventually burnt out by continuous antigen exposure and a hostile microenvironment, leaving the tumor partially controlled but not eliminated. Because of this, TIL therapy does not invent a new response, it rescues and amplifies one that already exists. So how does TIL therapy work? A. We have tumor resection. A piece of tumor is surgically removed, often from melanoma, but increasingly also from other solid tumors. B. We have isolation and expansion. The tissue is minced into small fragments and cultured in high-dose IL-2. Under these conditions, T cells that were resident in the tumor, so the TILs, preferentially expand. Fragments that show strong outgrowths are pooled, and the TILs are further expanded to billions of cells in large-scale cultures. C. Optional selection. In some advanced protocols, TILs are tested for tumor reactivity, for example by co-culturing them with autologous tumor cells and measuring cytokine release. The most reactive populations can be selectively expanded to enrich for highly tumor-specific T cells. D. We have lymphodepleting chemotherapy. As with CAR T cells, the patient receives conditioning chemotherapy, typically cyclophosphamide plus fludarabine, to create space, remove competing lymphocytes, and to reduce suppressive cell populations. And E. We have TIL infusion plus IL-2 support. The expanded TIL product is reinfused intravenously, often followed by systemic IL-2 to support their survival, proliferation, and effector function in vivo. Because TILs originate directly from the tumor, they form a polyclonal population recognizing multiple antigens at once, including shared tumor antigens and patient-specific neoantigens. This breadth of specificity may reduce the risk of a single antigen escape that can occur with highly focused therapies like CAR T cells targeting only one surface molecule. As for clinical milestones and combinations, after decades of work, this strategy reached an important milestone in 2024 when the FDA granted accelerated approval to Lymphocyte or Amtagvi, an autologous TIL product for adults with unresectable or metastatic melanoma who had progressed after PD-1 blockade and BRAF/MEK inhibitors if indicated. This confirmed that amplifying a patient's own tumor-reactive T-cell can yield durable responses even in heavily pre-treated disease. TIL therapy also integrates naturally with other immunotherapies. Prior or concurrent checkpoint blockade, for example anti-PD-1, may enhance the function and persistence of reinfused TILs by reducing inhibitory signaling. Conceptually, future approaches may further engineer TILs, for example by adding CARs or modified TCRs, or combine them with cytokine modulation or microenvironment targeted therapies to help these revitalized cells exert their full anti-tumor potential. And third, we have the natural killer or NK cell therapies. Innate immunity goes therapeutic. NK cells are part of the innate immune system and differ from T cells in a crucial way. They do not require antigen presentation via MHC to recognize a target. Instead of a single antigen-specific receptor like the TCR, they constantly integrate a balance of inhibitory and activating signals on their surface. Inhibitory receptors, such as KIRs or NKG2A, sense normal MHC class I expression on healthy cells and transmit a do not kill signal. Activating receptors such as NKG2D, NKP30, NKP46 or CD16 detect stress-induced ligands, viral changes or antibody-coated cells and promote cytotoxicity instead. When an NK cell encounters a target that has lost MHC class I, so is missing self or upregulated stress markers, and the activating signals outweigh inhibitory ones, it releases perforin and granzymes and kills the target. This biology makes NK cells particularly attractive in cancer because many tumors downregulate MHC class I to escape T-cell recognition, paradoxically making them more vulnerable to NK cell attacks. NK cells can often be used allogeneically with a lower risk of graft versus host disease or GVHD than T cells, enabling truly off-the-shelf products. And then there are natural mediators of ADCC, so antibody-dependent cellular cytotoxicity via CD16, so they synergize well with tumor-targeting monoclonal antibodies like rituximab or trastuzumab. It is reasonable to ask, if NK cells are so potent, why do we need to give more? In many patients with advanced cancers, endogenous NK cells are numerically reduced, functionally exhausted, or suppressed by tumor-derived factors and myeloid cells. Donor-derived or ex vivo activated NK cells can overcome this by providing large numbers of highly functional cells, sometimes with favorable KIR-HLA mismatches that increase their alloreactivity against tumor cells. NK cell therapy strategies include A. Unmodified NK cell infusion. NK cells are isolated from peripheral blood, cord blood, or induced pluripotent stem cells, so iPSC-NK, expanded ex vivo and infused back into the patient, often with cytokine support such as IL-2 or IL-15 to improve survival and proliferation. These products rely on the NK cell's natural ability to sense missing self, stress ligands, and antibody-coated targets, and have shown activity particularly in hematologic malignancies like acute myeloid leukemia and lymphomas. B. We have CAR-NK cells. NK cells that are engineered with chimeric antigen receptors targeting antigens such as CD19, CD33, or BCMA. Here, CAR signaling is layered on top of innate recognition, potentially combining rapid, MHC-independent killing with the antigen specificity of CAR technology. Early trials suggest that CAR-NK cells can induce remissions in relapsed or refractory blood cancers, sometimes with lower rates of cytokine release syndrome and neurotoxicity compared to CAR T cells. C. We have cytokine-enhanced or gene-modified NK cells. NK cells engineered to express IL-15 or other supportive factors can persist longer in vivo and maintain higher cytotoxic function. Additional edits, for example knocking out inhibitory receptors or adding homing receptors, aim to improve trafficking into solid tumors and resistance to immunosuppressive microenvironment. Most clinical experience so far is in hematological cancers, B-cell lymphomas and leukemias, multiple myeloma and AML, where targets are well defined and accessible in the blood and bone marrow. However, trials are increasingly exploring NK and CAR-NK approaches in solid tumors, such as hepatocellular carcinoma, lung cancer and ovarian cancer, often in combination with monoclonal antibodies, checkpoint inhibitors or cytokine therapies to help them infiltrate and function within the hostile tumor microenvironment. Because NK cells are not MHC restricted, a single, well-designed allogeneic NK product can, in principle, serve many patients irrespective of HLA type, offering a major logistical advantage. If their persistence and trafficking can be further improved, NK-based therapies may become one of the most versatile off-the-shelf tools in the adoptive cell therapy toolbox. Now let's look at clinical applications and the current status of ACTs. Today, all three ACT approaches are at different stages of clinical maturity but already reshaping oncology. TCR-engineered T cells have reached the first regulatory milestone with the approval of afami-tresgene autoloid cell for metastatic synovial sarcoma expressing MAGE4 in HLA-matched patients, and multiple trials are underway in other solid tumors such as sarcomas, myeloma and selected melanomas, often targeting antigens like NY-ESO1, PRAME or HPV-derived peptides. TIL therapy has likewise encountered routine clinical practice with leafyloid cell approved for unresectable or metastatic melanoma after PD-1 and targeted therapy and is now being tested in additional solid tumors including cervical, lung and head and neck cancers. NK cell therapies are a bit earlier in development but rapidly advancing. Several allogeneic NK and CAR-NK products are in phase 1 or 2 studies for acute myeloid leukemia, lymphomas and myeloma with encouraging response rate, while iPSC-derived NK platforms aim to deliver standardized, scalable, off-the-shelf cell drugs that could be used across many patients. Put simply, TCR T cells and TILs currently lead the way for solid tumors, whereas NK-based approaches are especially attractive for blood cancers and as versatile, ready-to-use allogeneic options. This all comes together as a multi-layered attack. Cancer is not a single problem, but a moving target shaped by genetic diversity, continuous evolution, immune evasion via checkpoints and MHC loss, physical barriers like stroma and abnormal vasculature, and metabolic suppression driven by hypoxia and immunosuppressive myeloid cells. No single therapy can address all of these layers. Adoptive cell transfer therefore sits alongside monoclonal antibodies, checkpoint inhibitors, targeted small molecules, surgery, and radiotherapy as one component of a broader multimodal strategy. Each ACT platform brings its own strengths for specific malignancies. CAR T cells have shown the greatest success so far in hematological cancers, especially B-cell leukemias and lymphomas and multiple myeloma, where stable lineage markers such as CD19, CD20 or BCMA are uniformly expressed on malignant cells and can be safely targeted across the entire compartment. TCR-engineered T cells are particularly attractive for solid tumors, driven by well-defined intracellular antigens, such as the synovial sarcoma or certain myeloma, melanoma and virus-associated cancers, because they recognize peptide MHC complexes derived from oncogenic or neoantigenic proteins that antibodies and CARs cannot see. TIL therapy is best suited for highly immunogenic, inflamed solid tumors like melanoma and potentially some lung, cervical or head and neck cancers, where a pre-existing T-cell response is present but quantitatively and functionally insufficient. Amplifying these polyclonal neoantigen-rich TILs can convert a weak endogenous response into a clinically meaningful one. NK cell products, including CAR-NK approaches, are especially promising in acute myeloid leukemia, certain lymphomas, and MHC-low or MHC-defective tumors, where their ability to sense missing self and mediate antibody-dependent cellular cytotoxicity or ADCC makes them a natural partner for tumor-targeting antibodies. In practice, these cell therapies are increasingly combined rather than used in isolation. Checkpoint inhibitors can be layered on top of CAR T cells, TCR T cells or TILs to reduce re-exhaustion and prolong activity. Monoclonal antibodies can debulk disease, alter the microenvironment, or recruit NK cells to support ACT. Targeted drugs and radiation can increase antigen release and render tumors more visible to transferred cells. The clinical question is therefore shifting from CAR T or TCR or TIL or NK to which combination of ACT and other immunotherapies best fits the tumor at this stage in this particular patient. Now, what is the future outlook? From off-the-shelf cells to in vivo reprogramming. Where is the field heading? Several clear trends are emerging that could make adoptive cell therapies faster, safer, and more widely accessible. First, off-the-shelf allogeneic products. Allogeneic TCR-T, CAR-T, and NK cells are being gene-edited, for example with CRISPR-Cas9, to remove endogenous TCRs and adjust MHC expression, reducing graft versus host disease and rejection. The long-term goal is to move from patient-specific, weeks-long manufacturing to standardized cell drugs stored frozen and given on demand. Second, in vivo cell engineering. Instead of modifying cells ex vivo, viral vectors or nanoparticles could deliver CAR or TCR constructs directly into T cells or NK cells inside the patient, turning them into engineered effectors in situ. This approach could greatly shorten timelines and costs, but raises questions about precise targeting, control, and long-term safety. Third, smarter targeting and safety switches. Logic-gated receptors, for example AND gates that require two antigens, aim to increase tumor specificity and reduce on-target off-tumor toxicity. Built-in suicide switches or drug-inducible on-off systems provide emergency brakes for highly potent ACT products, adding an extra safety layer. And fourth, better integration with the tumor microenvironment. Future engineered cells may not only kill tumor cells but also secrete cytokines, chemokines or enzymes that remodel stroma, normalize vasculature and recruit additional immune cells. The goal is to convert cold immunosuppressive tumors into hot inflamed environments where both transferred and endogenous immune cells can function effectively. Taken together, ACT is evolving from simply adding more T cells to deploying precisely programmed cellular medicines designed not only to recognize the tumor but also to shape the surrounding immune ecosystem. To conclude, adoptive cell transfer therapies beyond CAR T cells, so TCR-engineered T cells, TIL therapy, and NK cell approaches show how flexible the immune system can be as a therapeutic platform. TCR T cells extend targeting into the intercellular world of peptides presented on MHC. TIL therapy amplifies what the patient's immune system has already started, especially against neoantigens. And NK cell therapies bring MHC-independent recognition and truly off-the-shelf potential. Together with monoclonal antibodies, checkpoint inhibitors, and classical treatments, these approaches are transforming cancer from a purely cytotoxic battlefield into a field of immune engineering. In our next chapter, we will step away from mechanisms and back into history, meeting George Schuyler, one of the pioneers whose work on monoclonal antibodies laid the groundwork for much of what we have discussed in the last 2 episodes. Before closing, I have a small request. If you enjoyed my content and want to hear more, please like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.